Hello, Willowbrook. This is the video notes on protein synthesis today. So, moving away from DNA specifically, and more, how do we get from DNA to what makes us us? So right now, after this page, you don't have to write anything down. So the title of these notes would be protein synthesis. So again, the main function of genes is to control the production of proteins. So proteins help determine the traits of an organism. So this little picture down here kind of spells it out for you. We have DNA. DNA makes the proteins, proteins build the cells, cells build tissues, all up to the different bodies of the organisms. There's a monkey, or not a monkey, a penguin with a wrench. Why he has a wrench, I don't know. So this here on this page, you don't have to write this down right now. Don't write it down. So the order of nitrogen bases along with the gene form a genetic code that specifies what type of protein will be produced. So a group of 3 bases code for an amino acid. It's called a codon, which is a building block of proteins here. So these kind of combination of 3 chunks of bases form that will tell what amino acid is going to be built. So protein synthesis. The cell uses information from a gene on a chromosome to produce a specific protein. So anytime you see the word synthesis, you're making something. So protein synthesis, you're making proteins, you're putting them together. And again, this takes place in the ribosomes in the cytoplasm. So this is where you need to start writing things down. This is where the notes really begin. How do we get from DNA to whatever it is we're trying to make? That process in between. So the cell needs a messenger to carry out the DNA code from the nucleus out to the ribosomes in the cytoplasm. So RNA looks like one side of the ladder. So if we kind of cut that ladder in half, you just have the one half of the ladder, you're missing the other half. So RNA has nitrogen bases similar to DNA, has A, G, C, so adenine, guanine, cytosine, but instead of thymine, it has uracil. So a little different. One big change in those rules there. So instead of A to T, we have A to U. So there are 2 types of RNA. We have messenger RNA, we call it mRNA, copies the coded message from the DNA, and then transfer RNA, call it tRNA, carries amino acids and adds them to the growing proteins. There's 2 different types of RNA and kind of where it goes. Let's sort of break this down into the most basic steps we can to kind of understand how we go from A to B here. So DNA unzips, so we have half of the strand, and the RNA bases match up to form RNA, kind of like that we combine or replicate. So the mRNA goes into the cytoplasm and attaches to the ribosome where it reads the 3-letter code for that amino acid. The tRNA carries amino acid, which matches the 3-letter code base to the ribosome and attaches it to the mRNA. Kind of where it keeps getting built and getting brought back to that ribosome to get built. The protein chain grows until the ribosome reaches a message that codes for stop. So again, these amino acids are the building blocks of proteins. So some proteins are just a few amino acids, some are dozens and different combinations, hundreds of combinations of amino acids put together to form one protein. So what can happen here is there's a lot of chances for some mistakes in this coding. Just like if you're kind of copying a book from one page into another. A mistake in one gene can result in the incorrect protein being produced during the protein synthesis. So mutations can be harmful, they can be helpful or neutral, like it doesn't affect anything at all. So it often depends on where the animal lives, its environment, and if that might help the animal or hurt the animal. It kind of depends on its environment. If that mutation will help, hurt, or just not make a difference at all. Okay, so I'm going to try to keep these as short as possible. Thank you guys for watching and writing these information down. This will be very important as we go through the unit. So I'll definitely go back and review those steps, how we make proteins from DNA through RNA and actually get our protein. Review those steps is very important to know. So thank you guys for watching, and I will see you guys along the way.